Hello, my name is Graeme and today I will be talking to you about recruitment of deepwater corals on the continental slope off Nova Scotia, implications for habitat distribution and population connectivity, supervised by Dr. Anna Metaxas. First, what are deepwater corals? They are sessile, colonial invertebrates, and each coral colony is made up of tens to thousands of clonal cells. This is a close-up of the polyps. Each one of these polyps is an exact replicate of the next, and they use these tentacles to filter particles from the water column. They grow slowly, about the same rate as your fingernails, and can live for hundreds of years, making them vulnerable to anthropogenic disturbance. They can reproduce asexually through budding or fragmentation, as well as sexually through dispersal of gametes into the water column. Corals form important habitat in the deep sea by providing structure to the sea floor. You can see all the redfish gathered around the base of these colonies of Primnoa residiformis, commonly known as sea corn coral. This is an individual of Paragorgia arborea, more commonly referred to as bubblegum coral, and is roughly 2 meters tall. Together, these make up the most abundant cold-water corals in Atlantic Canada. Where are deepwater corals? Deepwater corals have a cosmopolitan distribution and are found in all the world's oceans between 200 and 4,000 meters. However, they are restricted to areas of hard substrate as they require a solid point to attach themselves to, such as ridges, walls, or boulders. In this photo, you can see colonies of bubblegum coral attached to a wall, and in this photo, you can see sea corn and bubblegum colonies attached to a boulder on an otherwise featureless bottom. Corals generally inhabit areas with strong currents, which deliver organic material for them to feed on. Due to these relatively strict habitat requirements, coral populations are generally discontinuous and spatially fragmented. Coral vulnerability and importance in the ecosystem has led to protections around the world, as well as here in Atlantic Canada. The Northeast Channel Coral Conservation Area was established in 2002, and the Corsair and Georges Canyons Coral Conservation Area in 2016, both with the goal of protecting the densest aggregations of bubblegum and sea corn coral from the impacts of human activities. For sessile organisms with a discontinuous distribution, recovery from disturbance depends in part on population connectivity with other coral patches, as well as successful recruitment of juvenile colonies. Recruitment in sea corn coral is presumed to be low, as colonies less than 10 centimeters are rarely encountered. It is important to protect adult colonies, but we must understand larval sources and sinks in order to effectively protect coral populations. So, the questions remain: where is there successful recruitment of sea corn coral, and how are populations connected to one another? This is a map showing the continental shelf and slope just south of Nova Scotia. In 2019, we visited 2 sites aboard NOAA's Henry B. Bigelow and did video transects with the remotely operated vehicle Ropos to look at distributions of corals. These are 2 dive tracks, R2111, which began inside and crossed the northern boundary of the Corsair and Georges Canyons Coral Conservation Area, and R2117, which explored to the northeast of the Northeast Channel Coral Conservation Area. This is an animation of the dive track in Corsair Canyon. The dive started near the bottom of the canyon, around 600 meters, and ascended the steep vertical walls. The black line represents the dive track which the ROV followed, and all purple points represent one colony of sea corn coral, and the green arrow points north. Walls near the deeper section of the dive were densely covered with coral, and as the dive became shallower, corals became more sporadic and were attached to haphazardly dispersed boulders on flatter terrain. No corals were observed shallower than around 300 meters. This is the same figure as the previous clip, with depth intervals overlaying on the dive track, and size frequency distribution of sea corn colonies divided up into 100-meter depth intervals. On the y-axis we have number of colonies, and on the x-axis we have size classes divided up into 5-centimeter intervals. The mean and number of colonies measured for each depth interval is shown here. The first thing you can see is that colonies get larger as the water gets shallower, increasing in mean size from 16.8 to 47.1 centimeters. The next thing is that the 500 to 600 meter depth interval shows a large number of coral colonies under 10 centimeters, while no colonies under 10 centimeters are present shallower than 500 meters. This is a photo from a wall at 580 meters with evidence of recent sediment slumping. Here you can see thick colonies of sea corn coral with a line where large colonies abruptly stop. We believe this is where the sediment slumped off, bringing any colonies with it. This is a close-up of the image, and as you can see, there has been successful recruitment since the slumping. This is the first time natural recruitment has been seen at such high densities, reaching 8 colonies per meter squared. Recruit showed a unimodal distribution and ranged from 1 to 9 centimeters in height, suggesting ages of 6 months to 4.5 years of age, and continuous recruitment over that time period. These are photos of coral colonies from 419 and 366 meters depth. And are characteristic of the corals encountered above about 450 meters. You can see larger coral colonies inhabiting a boulder on a relatively soft sediment bottom. This population structure, with high recruitment on walls between 5 and 600 meters depth, with no recruitment in shallower water, may provide evidence of sexual reproduction. During the dive, the current was flowing strongly into the canyon and up the canyon walls. If larvae were traveling in this current, they may come in contact with and settle on walls in this depth range first, leaving less larvae in the water column and resulting in discontinuous recruitment in the shallow water. This is an early hypothesis, as oceanic current data has yet to be processed. The next dive, R2117, was in the northern section of the Northeast Channel and occurs just outside the northern boundary of the coral closure. This is an animation of the second dive. The dive started around 800 meter depth and ascended the gently sloping valley walls. Sea corn coral colonies were scarce in the deeper section of the dive, but became more abundant in the middle of the dive, around 700 meters. The colonies occurred on poorly sorted cobbles and small boulders and ranged in size between recruits and colonies about 80 centimeters tall. Colonies continue in these high densities until the ridge, where they are less frequently observed. During the last section of the dive, the colonies became very sporadic and infrequent. This is the same figure as the previous clip, with depths overlain on top of the dive track, and size frequency distributions are split up into 100-meter intervals. This time there are two y-axes. The left-hand axis is colony count and are the blue columns as measured from the scaling lasers on the ROV, and the right hand axis is the number of colonies estimated to be less than 10 centimeters as estimated from the video footage and are the yellow bars. As you can see here, recruitment is highest for the 5 to 600 meter depth interval with over 400 recruits, which is the same depth interval where recruitment was highest in Corsair Canyon. The black points and bars above the figures are the results of a maximum likelihood estimation, which was used to estimate a finite number of normally distributed cohorts within a mixture distribution. Imagine the central point is the mean of a distribution, and the error bars represent the size of the standard deviation of that distribution. As you can see, in deeper waters we see few highly overlapping distributions and more numerous, less overlapping distributions in the shallows. This could represent a shift from continuous recruitment in deeper water to discontinuous recruitment in shallow water, which could produce these observed peaks through pulses of recruitment. It may also be due in part to variability in post-settlement survival. In these photos, you can see a large number of recruits attached to small cobbles. As corals grow, they likely become too large for the substrate which they are attached to and topple over in the current. Toppled corals attached to small cobbles were frequently observed in the Northeast Channel. In summary, we have located two geographically distinct areas with high coral recruitment, one in Corsair Canyon inside the coral closure and another in the Northeast Channel just outside of the current coral closure. Maximum recruitment at both locations was observed in the 5 to 600 meter depth range. In the Northeast Channel, height frequency distributions indicate continuous recruitment between 6 and 700 meters and discontinuous recruitment between 4 and 500 meters. Height frequency distributions indicate continuous recruitment in Corsair Canyon at 5 to 600 meter depth and may provide evidence for sexual reproduction. Next step, plans for my master's project. This is a map of sea corn coral distribution in Atlantic Canada. It's a combination of records for ROV surveys and trawl catch data. You can see that the records are quite sparse. Research in the deep sea is expensive and logistically challenging, which makes it infeasible to survey large areas. Thus, we rely on habitat mapping and species distribution modeling to fill in the areas which lack data to predict coral distribution. Using the data collected in 2019, in addition to previous ROV and TrawlCats data, I would like to model and map the distribution of sea corn coral in Atlantic Canada. After I have a better understanding of coral distribution, I would like to use the newly discovered recruitment locations as endpoints in larval distribution modeling. This is a figure from Metaxas et al. 2019 of modeled larvae into Corsair Canyon. Here you can see the Northeast Channel, and here you can see Corsair Canyon. There is a small chance of connectivity occurrence over a large area of the continental slope, and small area with chance of occurrence of over 50%. I would like to improve upon this work with better bathymetric resolution, allowing connectivity predictions on a finer scale, as well as include these new recruitment data in Corsair and Northeast Channel in the model. I would like to thank Martha Nizinski, the chief scientist aboard the Henry Bigelow, as well as the entire crew, my supervisor, Anna Metaxas, all of my wonderful lab members, and of course the funding partners, NSERC, Fisheries and Oceans Canada, and NOAA. Thank you very much.